Welcome to the High Dimensional Podcast. In this episode, Aaron and I spoke with Alyssa Antonopoulos, Senior Director of Data Informatics for Real World Data at Natera. Alyssa has led data teams working at the forefront of bioinformatics and real-world data and evidence. She's leading work using LLMs and human experts working together to abstract from structured and unstructured clinical data and combine it with genomics, biomarkers, and other data from diagnostics. We had a really great conversation covering those topics. Plus, we got Alyssa's advice on how to lead data teams. It's very exciting. How's it going?
Good. How are you all?
Really good. I did a deep dive recently into what Natera is. I think I understand like 20% of it because although I have spent a lot of time working on clinical data, I feel like every time I encounter bioinformatics related stuff, it just like slides past my brain and like leaves no trace whatsoever. So I was like reading through a bunch of stuff about Natera and I was like, okay, like
sliding,
kind of get it, like, but maybe that's like a good place to start because I feel like so many people have kind of come up with the grand idea of let's get all the clinical data, let's get all the genomic and bioinformatic and all the how many omics are there? Like, like 50, right? Proteomic, yeah, all of these things, and like let's jam them all together into this one thing. And I feel like a big obstacle to that not a lot of people talk about is the divide between bioinformatics people, everyone else who has no idea what they're talking about or what bioinformatics people are talking about all the time. So I'm just curious, like, do you run into that a lot? Like when you start talking bioinformatics, do you see people's eyes kind of like retreat into?
Yeah, I think it really depends on your audience, of course. I will say every time we get into a conversation about Natera data, particularly, I'll call it the flagship test, which is Signatera, it needs to be repitched, re-explained, even to savvy medical and sometimes bioinformatics folks that are on the receiving end of the conversation. And having joined Natera almost 3 years ago now, I feel like it took me a good 6 months to sort of understand the monitoring aspect of molecular residual disease test. In terms of data folks, like within our org, working on data, or even handling our data, if that's the case for like an external collaboration, I found that just really solid data engineering, core CS, can sort of work their way around the heterogeneity of the data, but you do need like that medical context to make sense of it, which I'm sure is the case with pure clinical as well.
Can you go into a little bit about what Natera does? I think my experience with you guys was when my wife and I were pregnant with our first kid, we had a genetic test for a bunch of diseases. And that was my first encounter with the word. And I was like, what is this thing? And so that's my entire familiarity with you guys. And then I've gotten a little bit familiar with the MRD space the last couple of years. And so I know you're involved with that, but maybe just like an overview of like, what are you guys focused on as a company? And then how you think about real world data?
Yeah. So same, that was my first encounter with Natera. I actually didn't even realize it when I joined that my first non-invasive prenatal testing was Natera's test called Panorama. And we also did our carrier screening through Natera's Horizon test. And that's essentially where the company got started. So in women's health, being able to detect trace amounts of baby's DNA from mom's blood. And that same technology has now expanded us into multiple therapeutic areas where, of course, we expanded within women's health to, in addition to the non-invasive prenatal testing, do the carrier screening. What genetic diseases are you susceptible to because you may carry an allele in that disease? And then expanded into oncology, where we detect trace amounts of circulating tumor DNA within your healthy sort of germline blood. And what it can do there is potentially detect whether or not your cancer is about to recur, or the other use case is whether or not you're responding to treatment, where theoretically your ctDNA goes down or clears, you're responding to therapy. It doesn't go down or it rises, you potentially need to switch therapy. And we've also expanded that same technology in organ health, particularly in organ transplantation, where we can detect trace amounts of donor-derived DNA from the transplant recipient. And that kind of helps monitor whether you're acclimating to your transplant and whether it's working. So yeah, 3 main business units, we're internally organized.
That's a really good overview. And I have never had personal experience with any of this testing. Like, so you mentioned like the ongoing monitoring, how frequently? I mean, I'm sure it depends a lot on the use case, but like, are we talking people getting like new sampling like every week, every month, every quarter?
Yeah,
year?
it depends on your setting of care and oftentimes aligns closely with like a Medicare coverage plan, as we are covered by Medicare in multiple indications. But essentially what it looks like for Signatera, which is the MRD test, is that occasionally a patient will have a time point or a test performed in the neoadjuvant setting, which means before they've had their curative intent surgery. And then the Medicare reimbursement really kicks in after surgery. So usually a surgical time point and more frequent testing closely after surgery. So maybe monthly or every 3 months. And then it will start to separate a little bit and perhaps it'll become every 6 months. And the guidances are usually something like, covering however many tests within 18 months of your curative intent surgery. But on average, our patients have over 4 tests per patient. And often it's because they just started their testing recently, within the last year. And so we have some patients that have 10 plus tests.
So the frequency between or how often the tests are probably varies a lot, but like what's common?
Commonly, we'll classify patients as active having a test within the last 6 months. So usually we'll see at least a test every 6 months.
And so what types of real world data, EHR data, claims data are you bringing to like join up with the MRD test data? And like what sorts of things are you trying to do with the combined? I don't know, maybe like lay the foundation of like how you guys think about that work, what you're trying to accomplish with that.
Great question. So of course we have the test result data. So that will comprise a positive or negative as to whether or not you have MRD. And then there's also a quantitative value. So 0 means you're negative, but pretty much anything above 0 means you have ctDNA, and there's like this aspect of velocity and whether you're rapidly increasing or rapidly decreasing. We have the test requisition form, which is going to have a few select fields related to patient demographics and like their general cancer diagnosis. So what is their cancer type and subtype? What is their stage? When were they diagnosed and when was their curative intent surgery? And usually, I mean, anybody who's familiar with the oncology space knows that that data is not nicely structured in tables in an electronic health record. So things like the progress note or the pathology report are like the gold mine that you need to actually extract anything meaningful and draw outcomes or response to therapy connections to the genomic data or the molecular residual disease data. It's great when we say, this patient cleared ctDNA, they were taking Pembro, and they had a scan that shows complete response. It's like that now you really have a full picture.
Yeah.
So how excited have you been about LLM curation of unstructured data and structured data? Because that sounds like you probably aren't going to have to hire 900 abstractors a la other companies.
Yeah, our abstraction model has been small and mighty, really complying nicely with the FDA guidances that have been published to date. And I would say like data quality standing out as a pillar. Like we'll go snail speed to make sure we're known for having really high quality data. And we will scale as needed with temporary staff or augmented staff, but they all have to pass the same like training requirements that we have for full-time staff. LLMs are super exciting. And of course, we jumped on the opportunity to like bring a faster human, so to speak, into the loop and perform sort of initial extractions with large language models that we can then use. We call it, like sometimes people call it humans in the loop. I prefer experts in the loop to validate, verify, or correct if needed. And that's like a workflow that we're actively pursuing.
And are you building that yourself or are you using other off-the-shelf tools, or what have you like experimented with? And if you're early, that's fine too. Just curious, like what direction you think it's going to go.
Yeah, I'll say that medical record abstraction has so many use cases at Natera. So it is a core capability that we need in-house. With that said, I think leaning toward internal development is like where we're headed. But we have no shame in saying like we lost to a vendor. And so we are performing some bake-offs, and we are assessing all available models internally, as well as multi-model approaches to really figure out which workflow will prove the highest accuracy or F1 scores, or however we want to measure it.
And prior to using large language models to extract some of the information, and I'm imagining in the workflow here, you have kind of a target data model that you're trying to create that is based on like kind of the analyses, the set of analyses that you want to do from like an outcomes, efficacy, whatever standpoint. Prior to LLMs, you're like doing a lot of kind of like, NLP like things to like do like named entity recognition to like find, oh yeah, they are on this drug, and then I'm going to put it there, and then I found the date and I'm going to put it there.
Yeah.
How is it evolving?
Yeah. So to your first point, yes, we have a pretty nicely defined data dictionary, I'm going to call it, or attribute value pairings. Like we try not to get too caught up in data model, because from an end user perspective, everybody may want some different view of the data. Attribute value pairs, and that can change a little bit across tumor types and across disease areas. So, as a real world data program, we do support the other business units, and we do a lot of data abstraction for chronic kidney disease as well. And I would say the prior sort of workflow had a little bit of sort of like NLP, if you want to call, basic string search NLP. We'll use our
regular expressions are our favorite.
We'll use our LLMs database and the information that we receive on the test requisition form to like whittle down into the right patient population. A lot of the inclusion exclusion criteria that we need to get to the cohort of interest is thankfully based on that basic like cancer type, subtype, and staging information. And then a huge thing on top of all of this is the MRD test is performed on a on a whole exome. So any sort of biomarker driven inclusion exclusion criteria is readily available via our research use only variant calling that we've done on those whole exomes. If we need to know, metastatic colorectal cancer patients who have a BRAF mutation, we can do that with just regular structured data.
That's cool. And so you're pulling all of this into like a big kind of a semi-structured like key value pair dictionary. It's almost like JSON, or it is JSON maybe.
Oh gosh. Yeah. Well, the current case, like pre-LLM, let's just say, is, we'll work in spreadsheets. And the abstraction team is actually, they're experts in like medical encoding too. So for their values, for these attributes, right? They have value sets, and all of those value sets have some sort of medical ontology mapping, whether it's LOINC or, ICD. They will conduct that as a layer on top of the data abstraction. But for auditability purposes, we of course will extract some semi-structured data, like what was the source text from which I derived this label and this coding system. And all of that gets stored eventually from those spreadsheets into a database. The LLM workflow and the verification workflow or software that we're building has APIs that connect to those databases and do that work for us. It's tough to be patient and wait to develop that type of process. And I personally have been putting it on hold. I'm kind of like, oh, not yet. Let's not spend the money on, great abstraction software and great NLP capabilities to make it faster just yet. Let's do a great job at high quality data. And as we scale up, go from there. And I think just by chance, like the LLM world boomed, and I was like, okay, great. Like now we can do it. And that was just by chance. Like I didn't know it was coming. It took me by storm.
A lot of workflows were like getting to that point where like, this is not going to be sustainable anymore. And then just in time, the LLMs came and like pulled the bacon out of the fire, like right just in time for a lot of folks with abstraction and curation.
I mean, that is the abstraction like workflow. Like everyone I've talked to and asked about how they're doing this, it's like, oh yeah, we have clinical informatics people like doctors working in spreadsheets. And I'm like, and like I've not talked to. I mean, it is evolving now, but that's kind of the standard, I think.
Yeah, and we tried like the more traditional NLP or machine learning approaches that were, a whole suite of vendors were doing prior to large language model inclusion. And I think we just found that though the accuracy could be high for certain categories of data, it was almost too low for too many categories that it didn't actually improve the workflow or that turnaround time. In some cases, your abstractors may actually be spending more time getting distracted by what was extracted and trying to confirm it, and then going through all of the documents that are available to find the correct value. And that's our biggest sensitivity to like switching over to an automated workflow is, are we actually creating a larger burden for the abstractors than what it was like in the manual workflow that they know and were trained on? And, you know, they're all nurses and that's the life that they've lived.
Yeah.
With that decision, like, do you just ask them? Like, or do you use metrics like to see how long it's taking, or how do you like kind of make that call?
Yeah, for sure. We have metrics on the manual workflow. Like we know and we can estimate, you know, when a project can be complete based on a number of patients and that disease setting where maybe metastatic has a lot more going on, more outcomes, more treatments. Early stage, not as much. So we know we have those time studies. You know, the abstraction team needs to be willing and involved as end users providing requirements to this build out. And they are, and they've been asking for this type of solution for some time. So that's great to have their buy-in. And then the next step is essentially performing those time studies again with the improved workflow. Other metrics like accuracy, F1, LLMs as a judge, exact match are all things that we're tracking in trying to improve those extractions in a quantifiable way. And so we don't know for sure if like we get to an average of 80%, does that actually improve things? If 3 out of 12 categories are terrible and our abstractors are taking, 20 minutes to resolve that issue. So really at the end of the day, we need those time studies.
I'd love to talk quickly about LLMs as a judge, because I've used that as well. And I'm just curious, like what others, because I think theoretically to me, I have a very basic theoretical objection, which is like, you have to believe that that LLMs are different enough from one another that they would like catch each other's mistakes, right? Like you have to imagine that their like functions are different enough that, Claude is going to catch something that Gemini didn't, or even like, or like different temperature will be like, and so, but it does seem like it kind of works. Although again, like maybe my issue is like, I don't necessarily have the best ground truth all the time to like actually judge against. So it's like quite difficult to say an objective ground truth, but like, I'm curious, like what your experience has been with that whole
ground truth point is a great point. We are fortunate enough to have been doing this for a while, to have like amassed thousands of patients with really deep, high quality clinical data. And as I mentioned earlier, aligning with those FDA guidances mean that we are doing IRR scoring. We're having 2 abstractors look at the same patient. We're ensuring those accuracies when that occurs is extremely high. And so the current golden state of like humans are considered the golden state for abstraction. We have a good base to compare to. LLM as a judge is interesting. It took me a while to wrap my head around it. But first of all, we chose a specific LLM as our judge, right? And I don't know if other people are doing that or if they're playing around with having different LLMs be the judge. But we chose based on more business terms. So let's choose an LLM judge that does have pretty high accuracy, but maybe is cheaper than some of the other ones, maybe is faster than some of the other ones. Like, let's not slow down the workflow because of our judge. And then we also asked our judge to, scale responses on a continuous variable 0 to 1, whereas exact match is very binary. Like, yes, it matches, no, it doesn't. And so there's a key difference between comparing, exact. And what it's great at is like in the case of LLM extracting triple negative breast cancer instead of malignant breast neoplasm, which is our label for breast cancer, it's not wrong. They actually do happen to have triple negative breast cancer, and it is mentioned in their chart. But exact match score is 0. LLM judge score is maybe like 0.7 or 0.8 or something along those lines. So it at least gives you a sense of like, okay, is the LLM actually like clinically incorrect, or have we not, tweaked it enough in prompting or whatever may have you in mapping that outcome to a label? And in in our example, our LLM judge like helped us narrow in on that. And it also helps us score ourselves a little bit more leniently on dates, where dates are like, in my experience, always the hardest. And in oncology, you have so much nuance. You know, how do you define the last date of therapy? Is it the first day of your last cycle? Or is it the last day that you went in and had your IV injection, you know? And perhaps different clinicians define that differently, but we define it a certain way. And you can sometimes see that the LLM is like a week off, and you're kind of like, okay, you know, you look through the chart and you're like, oh, maybe that was the last day they sat in the chair and received their dose, but it's not necessarily how we defined it. And it kind of helps us know that, okay, we're not like totally off. It didn't choose like a random date 10 years ago.
Has FDA not given guidance around that sort of thing, around dates?
Like the right way to kind of clinically define something?
Yeah, I guess there's probably enough variation depending on the analysis you're doing that maybe they couldn't.
Yeah, I don't know. But to your point, data dictionary flexibility and, you know, going beyond just like start and stop of therapy, but having more data attributes around cycles and dosing and things like that can help you hit more use cases depending on
and from a data, kind of back to the data structure thing, you go with the dictionary approach for flexibility, because then it's just like, what information do I know about this patient? I'm loading it into their massive dictionary. And then when it comes to actually doing the analysis, are your researchers kind of pulling from there into like R, into like Python, and they're like kind of building their own like data frames depending on what the analysis is that you're doing?
Exactly. Yeah. Our real world evidence team is typically in R, maybe Python, a few of them. Other, maybe more like commercial teams are using business intelligence tools. So as long as the data is nicely structured in a way that QuickSight can absorb it, you're fine there. Honestly, we have found, going back to the data modeling thing, to be almost unnecessary for a lot of use cases. The best need for it, in our opinion, in our experience, has been for delivery to an external partner and just having like some organization around the data. But at the end of the day, our internal real world evidence analysts just want like a long form table with like, you know, a patient hash, a test hash or sample hash, plus your data dictionary element, the value, and then any other information, metadata around it, units, et cetera. So it's super long form, and they can ingest that into their data frames and do their lollipop plots and all of that fun stuff.
Well, you heard it here first. Data modeling is unnecessary. That's the whole quote.
Oh, no. Or maybe it just like hasn't been addressed well enough in oncology. I think MCode was trying to tackle that, and like it's just always such a debate. Like oncology is a can of worms for sure.
Totally.
Well, weeding into that morass of challenging topics, like I guess would you say like I definitely hear you about the form of the data. Like people are pretty adaptable and they have certain things that they care about and other stuff that they don't care about. But would you say that like the semantic definitions are still really important to keep consistent? Or like, because I think that back to your point about dates, like, you know, what is the last day of the cycle? I'm curious, like, do you differentiate? Because I think some people kind of like those two things are the same to them, where they're like, I'm going to have a table and I'm going to define every variable very specifically, and then that's the data model, and then I'm going to populate it. But then a lot of times what you're actually worried about isn't the actual form of the data, but it is about the actual semantic definition.
Yeah. What I'll say on that is our data dictionaries, you know, don't only come with your usual like definition, example value, value sets, ranges potentially, but it comes with abstraction logic as well. So I guess even more specific to how to like therapy end date, last date of therapy or something vague like that. The abstraction rule section of the data dictionary will give you input onto why we defined it that way and how we prioritize source documentation. So for something like biomarkers, like we go to a pathology report first, and then we go to clinical notes if not available. And so try to provide as much context to the end users so that they have some understanding in the analysis that they're performing and can sort of apply rules as they see fit. against the data.
And is that embedded directly in the dictionary? Like that information, like you have the value, and then it's just like all of the metadata about like essentially the data curation process, when it happened, who did it, like all that stuff, so that the user has just acts. Because that's like one advantage of like kind of semi-structured like JSON or dictionary key value pairs is like you can just throw a bunch of stuff in there.
Yeah, it's not super belabored in the data dictionary that we might send an external collaborator. That like traceability of who performed an abstraction, when it was performed, what document they used, what the source text was, and things like that is all stored. But usually it is cleaned out of the ultimate data product that an end user is seeing. We're not like at a point right now where we have end users that want to analyze data based on like who abstracted it or something like that. We kind of keep that data quality control within the abstraction team.
What have been some of the analyses that your team has done that, I don't know, have been the most interesting to you or interesting in general in the space over the last couple of years?
Yeah, I'll give maybe 2 examples. So in oncology, we pretty much just started our real world evidence team and program. Natera has large translational medicine teams that have been pushing tons of scientific content and rigor into the public domain that have in large ways contributed to our Medicare reimbursement decisions. So I think a main pillar of Natera is like showcasing that scientific rigor and letting clinicians know that we have data backing the claims that we make. So that's Natera globally. The real world evidence team in our program basically just started last fall, and we have been on a roadshow, if you will, to derive insights from our real world data and get abstracts out there, start manuscripts, and just push some cool content that brings together the multimodality of the data. Such a buzz phrase. So one thing that we're doing right now in oncology is genomic landscapes, and then bringing in the ctDNA dynamics. So really understanding like the mutational landscape differences within a cancer type, across different subtypes, across different ancestry groups, across different biomarker subtypes, and then layering in ctDNA dynamics. And what I mean by that is, who had a favorable molecular outcome, who, remained negative throughout their journey, and who had positivity or who cleared ctDNA, who was positive and then cleared ctDNA. So layering that in with the genomic landscapes and potentially other clinical information like therapies they've been on is a big analysis that we are leaning heavily into on the oncology side.
Interesting.
Yeah. And then on the chronic kidney disease side, I'm super excited about is we are actually doing a lot of Kaplan Meier analyses. So progression-free survival analyses in chronic kidney disease patients and cohorts of patients. And there's tons of different genomically driven chronic kidney diseases out there that Natera is like a really good fit for, because we have a test called Renasight, and we essentially have a panel of almost 400 genes that we test against patients with a history of chronic kidney disease. And interestingly, CKD patients have a notion of staging like oncology does, where you can have stage 1, stage 2, and you can progress into end stage renal disease. But it's actually captured really well in EHR data. So
there's like distinct ICD-10 codes.
Exactly. And the labs like very closely align to your staging, like your EGFR levels, your proteinuria levels. So it's actually so much easier to perform those analyses on a larger scale in CKD. So we're bringing like a learning from oncology and how, progression and overall survival can like really dictate maybe decisions that you make in the course of your treatment or your journey. And we're like showcasing that in another disease area, which I think is really novel and interesting.
Now just imagine if we lived in a world where cancer staging just
seriously, except the debate is cancer staging doesn't change. So that is like a decision someone made a million years ago, and you're only staged at diagnosis, and then everything else is this really hard to find variable in like a scanned PDF somewhere. So
I would take all the fun out of it.
Probably a lot of jobs.
But the overall is kind of like the holy grail is like kind of like predicting outcomes, right? You have like all this super interesting information. What is the genetic starting point of the patient? What is the tumor, the tumor specific genetic profile look like? What treatment did they receive? When did they receive it? And then now you're like looking at the cell-free DNA to like actually see what's happening and just trying to, do you see a path where are you starting to see results where you can stratify the population and know who's going to have a better treatment effect or not based on those variables?
Yeah, I think so. I mean, in nature is a prognostic test, like, are you going to recur? And sometimes we can predict that just from your ctDNA positivity or Signatera positive months in advance of seeing a blip on a CT scan. And are you responding to therapy? And so hopefully moving from, okay, patient, is now positive, I'm going to scan the patient until I see some sort of tumor progression, to, okay, patient is positive from Signatera, I'm confident enough to treat now and treat early. And I think that is like the holy grail and what motivates me and what motivates a lot of people at Natera and hopefully in like the oncology space. And I think it's the same, in chronic kidney disease too, where it's not a monitoring test, but if you can understand what aspects about a patient makes them a rapid progressor versus like a slow or a regular progressor, to make those decisions earlier on and have better outcomes and, avoid the need for dialysis or transplant, like that's literally changing someone's life.
And that's just on the CKD side, that's just a difference in their genetic profile, like kind of their starting point
Right.
in terms of what's overall driving the disease.
Right. It's essentially a one-time test, very similar to, or we could draw a comparison to like your typical comprehensive genomic profiling on oncology, where maybe like a Foundation Medicine test or a Tempus test tells you, what genes you're positive for and therefore likely to respond to X, Y, or Z therapy. I'd say the difference with CKD is that we are where oncology was like 10, 15 years ago, where there actually aren't any approved therapies for your genetic cause of disease. All of those therapies are in clinical trials right now. But as those become approved by FDA, then soon you're in like that companion diagnostic space where, you're positive for APOL1 dual risk variants and you can take this drug and it's going to improve your outcomes. But for now, we're a little earlier, where we know patients with certain genetic backgrounds may be more susceptible to advanced stage of CKD.
Do you also pull in other types of more like classic clinical, like in addition to defining someone's starting point as their genetics, do you also look at their like comorbidities or like other disease history?
Outside of whatever we identified from chart abstraction, we also do work with third parties to kind of create that overall picture. So we'll pull in claims data, we'll pull in EHR data. I think with CKD, a lot of labs that you get tested on like a longitudinal basis create that picture. eGFR decline, for example, usually means you're progressing. For CKD, protein in your urine, we're creating that ecosystem. And we're also pulling in other types of outcomes like mortality, which, interestingly, a lot of our CKD patients aren't necessarily like dying right away because of their chronic kidney disease. And actually, same with our oncology patients. What's interesting about Signatera is it's actually like 70% of the time performed in early stage. So stages 1 through 3, where a lot of other genomic testing labs are in metastatic settings only. And so we have these patients like early in their cancer journeys, and they have, very strong and good survival statistics. And so we're seeing that really evolve over a span of many years. So our mortality data isn't like, we don't see a huge overlap necessarily, but as that builds up or as the data matures and as the test has been around for longer, we'll be able to start performing more studies and analyses that correlate, overall survival to ctDNA positivity or clearance as well.
Well, let's take the standardized oncology coding out of the picture. But if you could wave a magic wand, what would you, in terms of like, as you think about like what your dream situation would be for the future of real world data and real world evidence, like what things would you change? What would you change?
For me, I feel like no matter how advanced we get or however many technologies or vendors or offerings that are out there to aid people in data analysis, including AI and the black box associated with it. There just seems to be so much like post-processing. Like you can't just do a Kaplan-Meier curve. You need to have like choose your event, choose your starting point, calculate days in between, calculate your censored events. And it's just not something that's like out of the box ready to go from your starting point data set. And unfortunately, it just stalls innovation and like insights and evidence generation. I feel like that one is hitting home more this year because we want to make such a big impact in the scientific community, but there's just so much additional processing and even like curation and selection beyond the initial data set to get to an analysis ready cohort.
I was just going to ask, like, yeah, are those challenges like it sounds it's like choosing those, like choosing what time points to use, what you're going to censor, choosing your like, is that the type of thing you're talking about? Just like making those cognitive like overhead of getting people to agree on all that stuff,
Exactly.
documenting it.
It's so iterative. Like we define logic, we go with it. We decide, you know what? I don't think this makes clinical sense. Like we need to exclude patients with X and redo it. Let's take this event out of the censored events because it's like a broken arm surgery. It's nothing to do with their nephrology care or something like that, you know? And it's so iterative that you're lucky to get to a place where you feel like you have a code base in your repository that you can rinse, wash, repeat. So it's a little frustrating at times, but at the end of the day, you're just trying to make the most clinical sense and push out evidence that can like be backed by science in addition to like the data integrity.
And maybe as LLMs get better, it becomes less and less a problem of writing that, but there's still like the human element of actually like making the decisions of like,
yeah,
okay, like, do we want to define it like this? Oh, wait, we talked to Bob and Bob disagrees now, and let's go do it again. The cycle time maybe increases with LLMs, but the human discussion and decision making still has to happen.
And in addition to Bob, like we talked to ChatGPT. Does it make sense to?
Of course, yeah. Run everything through ChatGPT at this point.
Of course, yeah. That's just an added step to the process now. So I don't know.
You have to run it through 10 different models and have them vote LLM as a jury, you know, like
here's voting. And coming back to abstraction, maybe for a quick second, although I know we belabored that point. Someone mentioned to me recently whether like an LLM consensus approach, similar to invariant calling, often you have multiple callers and then you have like a consensus algorithm that chooses like the most likely scenario based on all of your inputs. I wonder if like consensus language model decisions is like an avenue that we should be exploring. It would cost a lot more, you know, run all 10 models and have a consensus algorithm, but would it be significantly more accurate? I don't know. We haven't answered that question yet. We've only answered whether routing different categories of data to different LLMs will improve accuracy, but not the consensus approach.
I've had a little bit of success with stuff like that.
Yeah.
So,
okay.
Yeah.
Something we'll explore.
We're kind of like rounding the curve here. And I was actually just thinking before this, you know, we all left Health Catalyst. We had a discussion where I asked you for advice about leading a data team because I was looking at that. You gave me some really good advice that I still reference every so often. And I'm curious, do you got any more? Because I definitely, definitely, for the audience, but also for anybody out there who's leading a data team and is looking for guidance on good practices for leading a data team, maybe also for how to talk to people outside the data arm of your organization about data, definitely I feel like would be of interest to some viewers and also me personally.
Gosh, I don't know if I can beat my previous advice that I don't remember.
Maybe it'll be the same then.
Yeah, it might be the top one that I referenced the most was be very decisive.
Yeah,
so
that's a good one.
That's a good one.
A luxury that I feel we did not have at Health Catalyst was having like strong product managers or like a specific software quality team, data quality team. Although we tried, carving out like specific roles there has been crazy improvement from Health Catalyst to Natera, where I think Natera puts a strong emphasis on like product managers that can really understand like the full life cycle of a product, and in this case, a data product, but also be like technical enough to help drive requirements with like an engineering mind. It's been such a huge difference. And I don't think that helps your specific question because you've got a data team. I don't know if you have product managers, but like really fostering that connection and collaboration with that team instead of like having any sort of resentment toward, oh, you know, they want to improve velocity or they change the requirements again, because that's always going to happen. So I guess I would just say like really working on those relationships, making them strong, and remaining agile and flexible will probably help.
Yeah, my words. Ellie. I mean, I think your point is excellent about, I think I've seen in a few different organizations, like different models of like engineers working with product managers. And it feels like something that's like really hard to get right, but also can be really great if you get it right. I'm curious, like, of that relationship, do you feel like it's sort of like you're able to have them help you scale, like talking to so many people? Because like think about data, right? Everybody has what they want the data to be, what data they want, et cetera. So like, is that kind of what the advantage comes from? Is if you're engaging with the product people, they're perhaps better positioned to go out and have all those discussions and consensus built?
Yeah, I do think that they bring that to the table. You know, a lot of our product managers are like PhD type scientists that can communicate strongly with medical staff, end users, as well as engineers. So that's great. But I don't think you should let that prevent engineers from having those conversations as well, because we've also had a ton of success in bringing our engineers in direct communication with the data recipient or the end user if they want to or they're willing to, or, you know, you focus on hiring staff that can do that. That's like a huge unlock, and it's a huge relief on the product managers too, that don't necessarily want to just be translators.
I've seen it all go the wrong way too, where it's like you end up with cloistered engineers that never talk to anyone, and just that's not good either.
That doesn't work well.
Alyssa, great to chat. We appreciate you coming on the show. Awesome to get the band back together.
Yes.
Wow.
It's been too long.
You have a reason to come to Boston ever, or still?
I might be coming for Nikhil's out of pocket data conference. I think that's in September.
Yeah, September.
I don't know. My plans are highly tentative, as always. But yeah, if it happens, I'll let you know.
True. Yeah, we can do a live recording.
Live round too. Yeah.
We need a backdrop and like cool.
Oh yeah. I'll go to Staples, get like one of those huge like banners that they put on restaurants.
Yes.
You know? Like, I'll be sick.
That would be fun.
Cool. This was great.
So much.
Thanks,
All right.
guys.
Thanks, y'all. Bye.